Okay, good morning, everyone. So the first talk is going to sound a little biological. That's not really the purpose, but I think for today's two lectures, I'm going to try to focus a little bit on the neurobiology of hearing, what the auditory system does. And then in the second lecture that Andreas will give, we'll try to give you a little bit more about how you use this basic knowledge that we learned from the brain into actual computational implementations. There will be a little bit of computational models that I'll talk about here, but Andreas will really get into a little bit more how this kinds of knowledge is implemented in actual engineering applications. So the basic outline that I'm going to go through, I'm going to just give you a little bit of some sort of few pointers about how the brain functions, how the communication between neurons happen. And then we're going to get into the auditory system, talk about the ear and the central auditory system. I will focus a little bit more on the auditory cortex because that's sort of the center of where auditory processing happens, but it's also a stage along the auditory pathway where we're just starting to learn a little bit more about processing at that stage. And then from that, I'll talk just briefly about how we can use this knowledge that we learn from the auditory system about robust representation of acoustic features that would be relevant for speech. And you'll see in this afternoon's lab that you can use a lot of this knowledge for a lot of applications. So the basic inspiration from all this work is that we have this brain. It does a lot of things that we would like to be able to implement in machines. It's able to recognize speech. It's able to do so robustly, even in the presence of background noise. There are a lot of things that it's able to do, and we're trying to really understand, well, how does it do it so that we can implement it in machines? That's the basic spirit of this whole approach. So I'll start first just telling you a little bit briefly about the brain. I'm not a biologist myself, but I'll just try to give you sort of an idea of how the neural structure, neural communication goes, and then get into the auditory system. So the brain is this sort of part of our body that is structured and has multiple, I don't know if I have a pointer or, all right, well, it doesn't matter. So we have it's constituted of a lot of nuclei. Each of which has a specific function, and people who study different parts of this brain try to understand what each part does and how the different parts communicate with each other. So as I just mentioned, it's divided into regions, and each is associated with a particular function. So you have the part that controls our motor actions, the part that controls our somatosensory, the visual cortex, and right in the middle there is the auditory cortex, which parses the auditory information. The language centers are spread between part in the auditory system, part in the association cortex, and in the frontal areas, because there is a lot of integration between what the acoustic cues come in our ear, the information that we already know about language, what we expect to hear, and all these things. And so there are a lot of brain regions that are involved in our perception of sounds and speech in particular. I will be focusing about the auditory system, none of these higher auditory areas, because that's basically the front end that takes the acoustic signal and starts parsing it. So just talking in general about the brain, so it is made up of neurons. People estimate that we have about 100 billion neurons in adults. So neurons are the basic cells that communicate with each other and constitute the brain. And they are very complex structures. So each neuron is connected to about 1,000 other neurons. Typically, each have many inputs, but they usually have only one output. And it's really this intercommunication between these neurons and the way they communicate with each other, the way they connect with each other, that constitutes this sort of complex neural networks. And that's really what the underlying processing capability of the brain comes from. And that's where the challenge is, because you have these individual elements, you just put them next to each other and have them communicate in a certain way. And that's what makes us intelligent beings that we are, and able to do all sorts of things. So this is just a diagram of the neuron's anatomy. So each neuron is made of a cell body. That's the main sort of what is called the soma, which is the zone where all the information that comes to the neuron gets integrated. It's made up of dendrites, which is where the inputs to this neuron come from the other neurons that are talking to it. So you have the dendrites that get integrated into the cell body, and it typically has one axon, which is its output wire. So it has multiple wires that bring in information, gets integrated in the cell body, and then the axon is the output of the neuron that gets sent to another neuron, to its own, one of its dendrites that gets eventually integrated into its own cell body. The communication between different neurons, this portion where the axon sort of connects to the next neuron, is called a synapse. So it's just sort of a little bit of terminology there. So this is not true of all neurons in the brain, but most neurons of the brain are like this. They have multiple dendrites, integrate into the cell body, and send the information along an axon to the next neuron next door. Some axons are very short because neurons communicate on a short distance, and others are very long, sometimes to bring information from one part of the brain to another part of the brain. So, so the interesting question is, how do they communicate with each other? And that's this notion of action potentials or spikes. So basically, you have impulses that come together from these different dendrites, get integrated into the cell body, and this leads to the generation of an electric pulse. As you see there, they get sent along the axon to the synapses that will send the information to the next neurons downstream. The interesting thing about these electric pulses is that for each neuron, they are fixed in amplitude and in shape. So basically, they send the same electric voltage. The only difference is that they can send it with different frequencies in time. They can send 10 per second. They can send 100 per second. So it's really this temporal information that encodes whatever it is the neuron is trying to say. They don't change the amplitude. They don't change how they are in terms of how the electric pulse is. They just change how frequently they send the information. So it's like a binary code. And that's how the whole brain communicates with each other, through these binary codes. They either send a spike or they don't. So, and this actual, what I'm calling spike, this action potential, is this electric voltage. It's often called an action potential, a nerve impulse, or a spike. So these three words are used interchangeably. I actually put this. If you have any questions, just feel free to interrupt me at any time. Okay. So let's look a little bit about how these action potentials are generated. So this is a sort of the inside and outside of a neuron. This is a cross-section of a neuron, just a schematic. And basically, neurons, just like many cells in our entire body, they have different concentrations of ions on the intracellular and extracellularly. Thank you. I can use this. So we have a high concentration of potassium inside, a high concentration of sodium outside, and we have these channels, these pores, that send the potassium out and bring the sodium in. And that keeps on going for each neuron. Now what happens is, if there is a stimulation that's sent in, I don't know if I can play this. Okay. So if there is a signal that is sent in, what happens is that these sodium channels open up and they bring the sodium in, which makes the inside of the neuron has a highly positive charge. And so you get this high positive charge that goes up. That gets counterbalanced with with potassium that gets sent out of the cell body. And so that brings the voltage down. And depending on the amount of sodium that comes in, if it exceeds a certain threshold, then this potassium gets counterbalanced, and that's what an action potential is. This is basically the spike. So you get the information come in from the external stimulus. So basically, you listen to a sound. A particular neuron gets disturbed. This sort of balance between electric voltages across the cell membrane gets disturbed, and so the potassium starts pouring in. If that amount of sodium exceeds a certain threshold, then the potassium channels try to counterbalance and bring the ions out. And that's how this action potential happens. This whole operation takes about 2 milliseconds, so it's pretty quick. So now once an action potential is fired, let's see. All right, so how do they propagate? So now we have an action potential that gets generated. This is along the axons of this particular neuron. The way they get propagated is that as the action potential gets generated and this counterbalance between sodium and potassium channels, the sodium channels next door get also excited a little bit. And so more sodium starts coming in, potassium starts coming out, and that generates a spike. So the spikes just propagate by having these different channels open in and out, with the ions going up and down. And that's how the spike propagates along the membrane until it gets to the next neuron, sends the information in, and then the same thing happens. The reason it doesn't propagate in the other direction is because when these sodium channels open up, they need time before they can open up again. And so by that time, the electric signal has already gone in that direction, and it doesn't go back in the opposite direction. Okay? So, and obviously, as you can imagine, any dysfunction in this whole operation, that's what causes a lot of neurological problems. If this propagation is not going through properly, then that causes problems in communication between these neurons. And that's why we have a lot of diseases come from these basic operations falling at one point or another. Okay? So just a term that I think is relevant, because I might mention it again, is this fact that the sodium channels take time. Once they open, they will take another time before they open again. That's called a refractory period. Okay? So, all right. So the next thing is, now that the action potential has been sent through this axon, how does the information get sent to the next neuron? And that's what is called synaptic transmission. As if you recall, we call this junction between two neurons a synapse. And so the basic idea is pretty simple. This is a chemical process. It's not an electric process. When the action potential comes from one neuron, it sends out certain chemicals, and those get transmitted to the next neuron. And that's how the signal propagates down to the next neuron. So it's pretty simple. I think I had an animation that shows, yeah. So the electric pulse is propagated along the membrane of this. So the sending neuron is called the presynaptic neuron. That's the one that sends the information. The other one is the postsynaptic neuron. It's pretty straightforward. Then you get this voltage that comes along this axon, sends these calcium channels. They make them open. And these, what is called the neurotransmitters, these are these molecules that get transmitted into the synaptic cleft, this part that connects the two neurons. And then the information comes through these receptors, and that's how the signal transmits to the next neuron. Okay. So it's a fairly simple operation, as you can see. It's, there's an electric pulse coming in, sending these neurotransmitters, the information comes out. Now, I guess when you look at this and see how simple it is, you wonder how we are able to do all these things with our brains. It's pretty simple. You just take two neurons, you get this communication through them this way, and then you just connect a whole bunch of them, 100 billion of them, and then that makes a human brain. So the question that we're more interested in is how is the brain, at least the part that deals with sounds and speech signals, how is it that it can do what it is able to do just based on this basic structure that I just talked about, on these basic neurons talking to each other? Okay, so the auditory system is made of a long pathway. It starts at the ear, obviously. This is where the acoustic signal comes in, and it goes through a lot of stages all the way to where perception happens. So each one of these stages has a specific function, and that's what people who study the neuroscience of audition or the neurophysiology of audition try to understand, what each stage is doing. The thing that we're mostly interested in as what I would call engineers, or somebody who's trying to use this information to implement it in in computational models is what are the features, what are that the brain cares about in these acoustic signals, what does it encode them and how does it encode them? Because that's what we're trying to really implement in these computational models. So, okay, so we'll start at the first part. So the ear, this is basically the organ of hearing, just like our eyes is the organ of vision. And so the basic operation that the whole ear, so one thing I should say is that technically the scientific term, the ear, does not just mean this part, but it means the whole organ all the way to what is called the cochlea, I'll tell you about. So this whole portion is what the scientific term, the ear, refers to. But commonly, when we think of the ear, we just think of the pinna or the external part of the ear. So, but what this whole organ does is it transfers the acoustic information, sort of the pressure waveforms that come from the acoustic signal, into an electric pulse. That's the language that the brain understands, these electric pulses. So this is where this transformation happens. You have the acoustic pressure that comes into the ear, and a transformation that happens here that sends these spikes, these action potentials, to get the processing started. Okay? So, all right. So we'll just talk about the three parts of the ear. It consists of three parts, what is called the external ear, which is what I just talked about, a middle ear, and the inner ear. So I'll just tell you about these different parts of the ear briefly. So the external ear is basically what plays the part of the acoustic antenna. It diffracts and focuses the waves coming in from the sound that we're listening to. And the ear canal acts as a resonator. And at the end of the ear canal, we have what is called a tympanic membrane, or an eardrum, that basically just, as the waves come in, this eardrum vibrates. Okay? So, all right. Okay, so next is the middle ear. So now once this eardrum vibrates, the middle ear is made of these sort of bones that move as the eardrum vibrates. So as the eardrum vibrates, these bones start moving, and they start pushing against some fluid that we'll talk about here in the inner ear. So it's basically this transformation from this mechanical movement into this motion of this fluid. That's what the middle ear does. The reason it has an important function is because it acts like an impedance adapter. So basically, the energy between the mechanical motion and the fluid motion, if we didn't have this middle ear, will get lost. There will be a lot of dissipation of energy. And that's what the middle ear takes care of. Okay? So, because keep in mind, outside here is air, and inside is a fluid. So you want to counterbalance the pressure from both. All right. So next is the inner ear, and that's what a lot of people think is the most interesting part of this organ of the ear. So it consists of a cochlea, which I'll show you in a little bit, and its basic function is like a frequency analyzer of sound signals. So as probably most of you have done at some point or another, when we take an acoustic signal, be it speech or anything else, we do like a Fourier transform on it to look at the frequency content. So this part, the cochlea, does this Fourier analysis in a way or another. So I'll just show you how that's done. So this is a basic structure of the cochlea. I think I have a real, this is an image of a real cochlea from I think this is a human baby. So you see it's this sort of snail-like structure. It typically has 2.5 turns. And the sound comes in inside this tube and propagates along. So now the interesting thing about this cochlea is that it doesn't have the same density from its base to its apex. So the base is the entrance of the cochlea, the apex is the end. And this is how the brain is able to do this Fourier analysis. So the way that's done is basically when the sound comes, all right. So when the sound comes, it propagates along this cochlea, as I just mentioned. So it comes in, there is a fluid inside this cochlea, and that's how the propagation happens. But the way it happens is that the sound wave comes and it stops at a certain point, depending on what the frequency of that sound is. So if you present single tones that just have 100 hertz, 500 hertz, 1 kilohertz, so if, say, I'm doing a 1 kilohertz sound, it will come through this cochlea, travel all the way, and stop here. If I give a 5,000 hertz, it will come and stop here. So this is how the Fourier analysis is done. Now, if I give multiple sounds with multiple frequencies, you'll have this part moving, this part moving. I think there was a little, yeah. So basically here. So if I have a low frequency, 100 hertz, it's going to travel almost to the end. 1,000 hertz will travel up to here. 10,000 hertz will only start and then stop. If I have a complex sound that has multiple frequencies, then you'll see motion in multiple parts of this cochlea. So as I'm talking now, because I have a certain frequency for my pitch, I'm saying certain words that have certain phonemes, that have certain structures, your cochlea is moving in different parts and doing this frequency analysis for you. So the location of where the cochlea is moving tells you what the frequency content of the sound is. And that's how the brain knows that I just spoke with, say, a pitch of, I don't know, of 200 hertz or something like that. Okay? Now, so this cochlear analysis is pretty interesting. The other interesting thing about it is that the frequencies are not aligned in a linear way. They're aligned in a logarithmic way. So basically, a step from here to here or from here to here is not, they're not equal in linear terms. So how can I show you this? Okay, so basically from here to here we traveled from 20,000 to 7,000, but from here to here we're traveling from 7,000 to 5,000. So it goes in a log scale. The high frequencies don't spend a lot of time, it does not put a lot of resources on the high frequencies, but it has more space on the lower frequencies. And that's why a lot of the models, the front-end analysis that do like the Fourier analysis, try not to do it on a linear frequency axis, but rather on a log frequency axis, because a lot of the information that is crucial in a lot of natural sounds, like speech, really lives in these lower frequencies. So you don't want to give more space to higher frequencies if you know that the information, there is no information that is relevant for your perception there. Okay? All right. So, okay. So there are two more important components in this cochlea that are worth talking about. So in the center of this cochlea is what is called the organ of Corti. We're not going to go into the details of this biology, but the interesting part is once these fluids start moving inside the cochlea and stop at a certain point, we have what is called hair cells on the edge of this membrane that start moving with this fluid. And this is how we know the motion is happening through the cochlea. And it's the motion of these hair cells that gets transduced into an electric pulse that is sent up to the brain. So this is why this part is important, because this is where the transformation happened. From the mechanical motion to the electric pulses, we have two types of hair cells, what is called an inner hair cell and outer hair cells. Just have, I think, one slide. Yeah. So the inner hair cells have these, what is called like hairs or cilia. These cilia move. This is a real picture of a hair cell. So these cilia move with the motion of these fluids inside the cochlea. And the point where the motion stops is where the frequency of the sound is. But we see that these cilia can move with a pretty fast motion. And the transformation from this motion here into an electric pulse is what sends the signal down to the brain. The only thing is that these hair cells are not neurons. They don't send electric pulses themselves, but they send these synaptic, like we saw, the synaptic transmission. They do open up a gate. They don't send an electric signal along them, but they open up the gates, they send the neurotransmitters, and that's how the signal gets propagated down. That's all I'm going to say about them. We're not going to get into details. The outer hair cells are interesting cells because the way they transmit information is by changing their length. So what is called hair cell motility, so they become longer and shorter. You can see here through this baseline. So as the sound comes, they become longer and shorter. And their job in the brain is to basically increase our hearing sensitivity. They amplify the quiet sound. So by moving up and down, they make the membrane underneath move. And that's why even when there are very faint sounds, we can hear them. Because by moving that membrane, it creates it gives us the sense of the presence of a certain sound, even though it's pretty quiet. They also close the loop, so they get a lot of information back from the brain down to the ear that gets sent to them, and that balances our hearing for very loud sounds and very quiet sounds. So they do a lot of feedback control. I just wanted to, it's not really relevant, but something that you should know about is that losing these hair cells means that we lose our hearing because they're the ones who transmit the information. And so exposure to very high sounds, rock concerts, very loud music, kills these hair cells. So you see here, this is a cat that was exposed to very loud noise for 2 hours. And you see its hair cells. So we have a whole bunch of hair cells, some long ones, some short ones. This is just exposing a cat to 2 hours of very loud music. Or I don't know if it was music, maybe it was just noise. But you see after the exposure, the long hair cells have all been killed and mushed with the shorter ones. And that's how people lose their hearing, because these hair cells get destroyed. They're pretty sensitive. So by losing them, that's how we lose our ability to hear. OK, in any case. So next thing. So this is a schematic of the auditory pathway that we're going to be talking about. This is the cochlea that we've been talking about. Once the signal gets transmitted from this cochlea, then it travels into a lot of pathways all the way to what I call the auditory cortex, which is the center of where the processing and sound perception happens. So we have multiple stages. There's a cochlear nucleus, which is the first stage that this auditory nerve sends information to. Then we have a whole bunch of other stages. I'll just tell you briefly about what each one does. So typically when we try to study how the auditory system functions, we try to understand what each stage does and how the stages communicate with each other. For us, because we're not biologists, we're not trying to understand really the sort of the neurophysiology of how the transmission happens, but rather we just want to know what features in the acoustic signal does each stage extract, and how does it send it to the stage next door. Because when you're trying to really come up with a computational model of how this operation happens, you want to know, is there a part of the signal that I should not care about that is irrelevant? What is the part of the signal that carries most information? If I'm listening for speech, do I need to encode the whole speech signal? Do I need to really care only about certain features in that speech? And by knowing what the brain cares about, I know that this is what I should implement in a model. And I don't need to care about the other parts. So the cochlear nucleus, the first stage that comes after the cochlea, it has multiple branches, a dorsal and a ventral one. And it plays multiple roles. It plays a big role in detecting sound onsets. It has these broadly tuned cells that don't care about frequency, but just cares whenever the onset of a sound happens. And it also plays a role in detecting spectral notches. We don't need to care about the integration of somatosensory information, but it also plays a role in estimating sound elevation. So as you might notice here, I'm telling you about, we have 2 ears. And one of the big reasons we have 2 ears is to allow us to hear sound in space. So when you're listening to me, you know I'm standing right here and not over there. And the way you're doing that is partly because you have 2 ears. And you're using this information from where the sound is coming to your ears. The fact that I'm talking from you, say, for you it's more direct, but for you it's more on the side. So as the acoustic waveform travels all the way to your 2 ears, they come at a slightly different time in between this ear and this ear. They also come at slightly different loudness because of reflection from your head. And that's what the brain uses to know that the sound is coming from this way. So this integration starts a little bit, this information starts being extracted here, but starts really get integrated at these middle stages, this acoustic information from space. Okay? Okay, so the next stage is this superior olivary nucleus. It It's nuclei, really, because it has multiple parts, obviously. Nothing is simple in the brain. But the basic idea is because it plays this one plays a major role in extracting this acoustic information, particularly for sound localization. And so an interesting thing is that the size of this nucleus varies between different animals and humans because we have different sizes of heads. So depending to an elephant, because it has a big head, the acoustic information to its 2 ears is going to take a longer time to travel. We have relatively smaller heads, so we have different time in between of information coming to our 2 ears. So depending on the animal's size, there's a lot of different information that needs to be extracted. And this nucleus has different sizes also, probably because the processing is different between different species. Okay? So next we have the lateral lemniscus, and that also plays a role in localization, in extracting spatial cues, and particularly the timing of echoes of this thing that I just mentioned, this interaural timing differences between the 2 ears. The time that the signal comes to ear 1 and ear 2 is going to be slightly different on the microsecond level, but it is where the information about where the sound is coming from is. So that's part of the brain that tries to extract that information. The next stage is the inferior colliculus. That's what is called the midbrain. So we have a peripheral stage, the middle of the brain, and then we have the cortex, which is the central part of the brain. So at the midbrain level, all the information, all the cues that have been extracted converge to the midbrain. So it puts all the information together and sends it to the processing, to the CPU. So the information gets extracted, and then the CPU takes all that information in and says, OK, this is a female voice saying so and so. OK? So it extracts all this information. It does a big job in doing for sound localization, as I just mentioned, but also encoding specific amplitude modulations, pitch information. Next, we have the auditory cortex. That's what I've been mentioning as the CPU. I'll tell you a little bit more in details about it in a little bit. But the basic thing you need to know about the auditory cortex is that it's very important for processing temporal sequences, like speech, for example. So we know, for example, that when people do experiments in animals where they remove their auditory cortex, then they have a harder time discriminating between sounds that have different temporal structures. They don't have the processing part of their brain that can put sequences in order. And speech is all about temporal information. You have phoneme A follows phoneme B, phoneme C, and that order is very important. And the auditory cortex is the stage that does that. We also know that human patients with this bilateral damage, and they don't do these experiments in humans, but the humans who happen to have this condition, we know that they have severe problems in processing temporal order. And without the auditory cortex, then we don't really have an awareness of sound. We can react to it reflectively if you slap your hands or do something, then the person will react, but they don't really feel that they are aware there is a sound there. So that's where the awareness of sound really happens. Okay? So the auditory cortex has multiple stages itself. In the rest of today's lecture, I'll tell you a little bit more specifically about the primary auditory cortex. That's the first stage of the CPU operation. What we know from it is that it's a pretty complex structure, but it encodes some important features that we're going to talk about in a little bit. One of the interesting things about the auditory cortex is that it has a precise topographical map. So what a topographic map means, and in the case of the auditory cortex, we call it tonotopic map, means that neurons that sit next to each other do information in an orderly way. Just like we saw in the cochlea, you have 1 kilohertz followed by 2 kilohertz followed by 3 kilohertz followed by 4. The same thing in the auditory cortex. Neurons next to each other encode, for example, frequency in an orderly way. When you go to the secondary auditory cortex, it doesn't do so. It just has neurons that do, you can find 1 kilohertz next to 100 hertz, next to 5,000 hertz. There is no order in that. But at the level of the auditory cortex, that's still true. And one interesting thing about the whole auditory system is that this tonotopy, this Yeah, I had it here. The word tono comes from Greek, which means place of tone. And okay, and so what I was saying is that most of the auditory system, from the cochlea all the way to the auditory cortex, so all these stages that I've told you about, they all have this function. They have very ordered topographic maps. So this is not, yeah, I think I had, yeah, here. So this is the figure I showed you earlier of the cochlea being organized with frequency organized next to each other in an orderly way. This is looking at the auditory cortex, and what we find is we find neurons that are also organized in a tonotopic way. So 500 hertz followed by 1 kilohertz followed by 2, 4, 8. And again, as you see here, you see the log scale organization of the sounds. So from the cochlea all the way through the stages in between, all the way to auditory cortex, you have this organization that is maintained. Yeah, I was going to say something. Okay, so this is just a little segue, but it's important to know that this topographic organization is not only unique to the auditory system. Other sensory parts of the brain also have it. So we have it in the visual system, we have it in the somatosensory, and there's also some evidence that we have it in the motor cortex. So basically, for example, in the visual system, as you record from neurons next to each other, then they represent locations in the visual space next to each other also. So if I go in the visual cortex and I take the first neuron, go to the next one, go to the next one, then I find that they are encoding information from this part of visual space, this. So they don't do it in a disorderly way, they do it in an ordered way. With exceptions, obviously, since this is a brain, so it's nothing as simple. It's also true of the auditory system, but you see this occasional discontinuity sometimes. But more or less, they do so. In the somatosensory cortex, we have the same thing, what is called sort of somatosensory topography. So basically, neurons in somatosensory cortex encode different parts of our body. So basically, you have the finger. The neurons that encode finger information are next to neurons that encode this finger, next to this finger, the arms, the shoulders. So it's all organized. So neurons that encode this information are placed next to each other. Okay, so back to the auditory system. So what I was saying is it has this precise topographic map, or what is called the cochlear map. So people use topographic map, tonotopic map, cochlear map. They all mean the same thing. Basically, neurons next to each other encode frequency information that is organized in an increasing way or decreasing way. Okay, so the other stages that I just will briefly mention is we have secondary auditory cortex. So from this primary auditory cortex that I was telling you about, it's commonly referred to as A1. Just like in the visual system, the primary visual cortex is called V1. In the motor cortex, the primary motor cortex is called M1. So auditory cortex is called A1. So we have A2, which is a more complex part of the CPU. We really don't understand well what it does. We know that it has some topographic maps, but they're not very well organized. We don't have this frequency very nicely ordered, or we have very small parts that are ordered, but as a whole, it's not very well structured. We know that it doesn't really respond well to simple sounds, like a boring noise. It will not respond at all. It will only respond if you give it more complex sounds. This is also the case, by the way, of the primary auditory cortex, A1. If you just give it a boring noise and keep playing it and record in the auditory cortex, it will just stop sending these action potentials. It's boring, it doesn't care, there's no information there, it will not respond. So, okay. And then we have the next stage is planum temporale. I think I'm just going to skip these parts, but the whole idea that I was trying to get you to is that you have these multiple stages, and then you start getting into areas where we have linguistic information stored, information about music, what we know about sounds. And that information gets integrates what you hear with what you already know to guide how your perception goes. And other stages play a role in this sort of whole integration. There is association cortex, there is the frontal parts of the cortex that play a role, but they're not technically part of the sensory cortex, the part of the brain that parses sensory information that comes from the outside. They store already existing information, so we're not going to talk about those. Okay, so now how do we know what each part of this brain does? So the way we do it typically is, I mean, you can do it multiple ways, but one of the most common ways is what is called single unit electrophysiology. So basically what you do is you put an electrode inside an animal's brain, close to one of these neurons, you play a sound to the ear, and you let the electric signals travel, and you record how the electric signal looks like. And then you try to do this sort of transfer function analysis. You have this black box, you know the input, you know the output, what happened in between. So, and you do this for every single neuron in each one of these stages. What we care about, as I mentioned earlier, is what frequency got encoded, what feature of the sound did this neuron care about, and that's how we learn about what this whole brain structure does. So I'm going to be telling you a little bit more about what the auditory cortex does and what features it really cares about. Okay. So as I mentioned, we have this sort of input-output mapping. You can go into this forward model where you can start from a signal, record the neural responses, and try to map this transfer function from the signal to the neural response. And we're trying to do this analysis of what the transfer function looks like. What would give me this transformation from signal to neural? Some people do what is called like an inverse model. You can try to guess what the sound, so if I give you blindly a neural signal, I tell you, can you guess what this neuron is doing? And you can try to reconstruct back the signal. The challenging part with doing this inverse model is that if I give you information about only one neuron, it's probably impossible for you to know what the sound is, because each neuron only encodes part of the information. It's a whole network that parses the full signal, but one piece of the information might be impossible for you to do the inverse model. So I'll tell you specifically about the case in the auditory cortex in A1, if you remember, and we're going to focus about just this forward model. We present sounds, we record the neural signal, and we try to measure this transfer function of the system. Okay? You're all familiar with what a transfer function is. Okay? Yeah? All right. So the way we do this is we present these sounds, as I said, we measure the neural response, and we try to characterize this transfer function. This transfer function is going to be called STRF, spectral temporal receptive field. So it's the transfer function, but in spectral temporal domain, because we care about the information both in time but also in frequency. So this is just like the way you do transfer function measurements in a linear system. So basically what you do is, let me do it here. So if I have a linear system with a transfer function h, I give the input and output, then in the most classic way, the way you do it is you present single frequencies, right? So if this is my frequency axis, I present this frequency here, and I measure what the output to this frequency is, right? I do this here for every single frequency, and that's how I measure the transfer function in the frequency domain, right? So we're doing basically the same thing. The only difference is we want to do it both in spectral but also in temporal domain. By that we mean that this is a spectrogram of each one of these signals. That means we modulate them in time. They have a certain modulation in time, but also a certain modulation in frequency. And then we vary these modulations in time, and we vary the modulations in frequency. So this is the only difference than doing here. The reason we do it this way is because, as I said, the auditory cortex doesn't care about borean sounds. And a single tone is a borean sound, so it will not respond. These sounds, on the other hand, so I'll play you one of them. This is what is called a ripple. So this is a noise that is modulated both in time and in frequency. So it has some structure to it. The auditory cortex likes to respond to these things. They have some information as opposed to just a beep. It just doesn't care. It gives a few spikes and then it stops. So the way, as I said, is we just vary this information. Just like you would vary frequency there, you vary temporal modulations, so you make them go faster or slower, and you vary the spectral information. So I'll play you these. So this is the same noise, it just modulated a little faster. And then you can vary also spectral information. It's very subtle. So you can hear it here. So it has the same temporal dynamics, these two. They just have different spectral content. And in theory, just like any sound can be decomposed into its frequency components with a Fourier analysis, the same thing is true of these ripples. They are a complete basis set into two-dimensional Fourier space. So each one of them is like a sinusoid that is modulated in time and in frequency. And I can decompose any sound to a sum of ripples. Just like I can decompose any one-dimensional acoustic signal, S of t, into its Fourier components, its frequency components, I can do the same thing in two-dimensional space. So this is the same idea. Now we just present each one of these ripples. They're called ripples. So I present each one of them, and I measure the transfer function. And the way we do it is just like you would do the transfer function in one dimensions, except that now we do it in two dimensions. So you convolve the response with the structure of the stimulus, and that gives you back the transfer function. Okay? So as I just said, so we present each one of these at one time. We measure what the neuron gives us. And the neuron, as I mentioned, what they give us is they give us these action potentials. They give us these electric pulses. And there is no information in their shape. There's only information in their time and how often they give us a spike. So this is what is called a raster plot. A raster just has each dot here is one spike, is one action potential, one electric pulse. And it varies because we're varying the stimulus. So when we give, for example, a 4 hertz modulated ripple, it gives us few spikes here, 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 here. And you can see more or less that it's following. It gives us a spike every 250 milliseconds. That's 4 hertz. It's encoding. It likes 4 hertz. It gives us a spike that follows the stimulus. The same thing here. We give it 8 hertz. Now it starts giving us a spike every 125 milliseconds. But not all neurons like all frequencies. So what we do is we present each one of these ripples one at a time, and we measure how well does it like this one, how well does it like this one. And by how well, we mean how faithfully does it encode this temporal modulation. Does it give me a spike every 250 milliseconds? Does it skip one cycle? Does it really not follow at all? And once we know for every one of these modulations, then we fill in this two-dimensional space. It likes 4, but it doesn't like 8. And we do it. And then we take the inverse Fourier transform. That gives us a time-frequency representation of the transfer function. Okay? So again, this is in the modulation space. This is in this space that varies temporal modulations and spectral modulations. And when we go back, then we see time frequency. And green is just the baseline response. Red is positive. Blue is negative. And I think I have examples here. So basically, these are 6 different neurons in the auditory cortex. And you see that each one does something slightly different. So for example, this neuron likes to respond to frequencies around, so this is between 1 and 4, so around 2 kilohertz. So what you see here is green means 0. So it does not respond to anything below 1 kilohertz. It doesn't care. It's not its job. Its job is really to encode information here. And the way it encodes it is with this temporal dynamics, with an impulse response that expands up to 50 milliseconds. On the other hand, this neuron encodes 2 kilohertz as well, but with different temporal dynamics. They expand all the way to 100 milliseconds. So what that means is that the temporal modulation, how fast a sound goes, one neuron would like a slower sound. This would like a faster sound. Okay? The same thing in spectral information. This neuron likes very broad, a lot of frequency content to sound, very broad sounds, while this neuron only encodes information around 2 kilohertz. So Yeah. So depending on what the signal is, you can know what each neuron is doing, but this is putting all of it together is how the sound is being encoded. Now, the way we map this into a computational model, and what is very interesting about the encoding of sounds in the auditory cortex is that this temporal dynamics.
Mounya,
Yes.
can I ask you a question? Sure. So let's say we take the top left plot, right, the first neuron.
This one, yes.
So does this mean that if I give this neuron a very short 2 kilohertz burst, then 10 minutes later it's spiking very rapidly, that's the red dot.
Yeah.
And then about 50 milliseconds later it's much quieter than normal, so it
Yes.
has noticed that the burst has gone away.
Mm-hmm.
And then after that it again gets back to being its normal state. So that's
Yes.
how I read this. So
Yes.
I imagine this as a burst at that frequency for a very brief period of time. And then this is how it's responsible with all over time. So it's an impulse both in frequency and time. Or do I assume it's a steady 2 kilohertz?
It is a steady, well, you can interpret it as the first interpretation, but because you know that this is auditory cortex and if you give it like a steady boring sound, it will not give you that. But if you give it a sound that has the same modulation, say at 2 kilohertz that has a modulation with these dynamics of 50 milliseconds, which is basically 200 hertz, is it?
Right.
Then it will give you a burst and shuts down, a burst and shuts down, following the pulse of your signal. So if you give it
So
this,
the signal is a AM signal.
an AM signal,
modulation is
yes.
200 hertz after 50 milliseconds.
Exactly. So it will give you this, shuts down, gives you this, shuts down. So it follows very faithfully that. If you give it a slower one, then it probably will not respond. So
But it would also detect an onset of a constant.
it will detect also so the first interpretation is also true. If you give it just a short burst with those dynamics, then it will match it as well. Okay. So now the reason all of this is interesting is because neurons in the auditory cortex don't encode any information. So basically, very fast modulations of the order of 100 hertz, you won't find neurons that really follow them. They will not give you a spike every 100 milliseconds. Neurons in the auditory cortex like more or less slower information of the order of 10 hertz, every 100 milliseconds of the order of 4 hertz, every 250 milliseconds. So they like slower modulations. And the interesting thing about that is that when you look at speech and the modulation content in speech, you look at, say, a modulation profile of a speech signal, then most its energy is really around these slow modulations of the order of 4 hertz, 8 hertz, 10 hertz. That's the sort of the syllabic rhythm by which we talk. We don't talk as fast as a sort of 100 hertz syllabic rhythm. We talk of sort of 10 hertz kind of order, 4 hertz kind of order, 250 milliseconds. And so this nice matching between the tuning in the auditory cortex and actual signals like speech is very interesting, because when we try to think of where the information resides and what the auditory system cares about, at the level of the auditory cortex, it seems to really care about these slow envelope information. The second reason why that's interesting is because if you distort a speech signal, you low pass the very fast information, you keep only the slow envelope, you can still hear it. It's intelligible. It doesn't sound nice because it's been low pass filtered, but it is intelligible. So because the information is really in these slow modulations. And the auditory cortex seems to only encode these slow modulations. That's where the information in speech is. You have the other stages before the auditory cortex that get the detailed information, but once you get to the CPU part, the part that puts things together and perceives speech, it's really only the slow modulations that matter or that it cares about. Okay, so now, all right, so we're almost there. I'm just going to show you briefly how we can take this information into putting it in, say, for example, a computational model. And in the lab this afternoon, you can work with this particular model that I'm telling you about and see how you can use it for doing different things. So as you saw earlier, we have this early part that does a lot of analysis of the signal, and then we have this at the level of the auditory cortex that does this further spectrotemporal analysis of sound. So the early stage, and this is just one model that simplifies these different stages. There are a lot of biologically inspired models out there that try to mimic this information about the auditory system that I told you about, and this is just one of them. So So this model, what it does is it parses from the acoustic signal to a time frequency spectrogram, like the Fourier analysis that we would typically do, except that now it tries to add some information to it. So for instance, instead of a linear frequency axis, it implements a logarithmic frequency axis, because we know that's important. And it also adds a little bit of other stages. So the first part it does is this cochlear analysis. So the acoustic waveform is a one-dimensional signal, just as a function of time. Now we need to do this frequency analysis that the cochlea did. And the way it does it is just with a basic sort of affine wavelet analysis. So we have these bandpass. I have this thing. So we have these bandpass filters. And then we do the analysis of the signal for each one of these bandpass filters to get different frequency channels. The only catch here is that these are not symmetric filters. They are asymmetric, trying to mimic the shape of how the cochlear analysis is done. I didn't put the equation for how the filters are implemented. But the basic idea is that they're also constant Q filters. Basically, you know what constant Q filter is. As you move from frequency, the bandwidth scales with frequency. So they basically get wider at higher frequencies or are narrower at lower frequencies. Okay. Then the next stage, we have this inner hair cells that we mentioned before. And the way that operation is done is we do a high-pass filtering. We have a nonlinearity because the operation in the hair cells is not a linear one. And then there is a low-pass filtering. Now, one interesting thing about this hair cell stage is that, if you remember, what I said is that the output of the hair cells that goes to the auditory nerve sends spikes. And it sends them relative to what the acoustic information is. So if you give it a 1 kilohertz sound, it will give you a spike every 1 millisecond, because 1 kilohertz is a cycle that has 1 millisecond cycles, right? If you give it 200 hertz, then it gives you 5 spikes per second or something like that. Now, if you give it really, really fast signals, I'm sorry, you can see, well, there's nothing to see, it's really just this. But so if I give it really, really fast, above 4 kilohertz, then those neurons cannot follow. They follow up to 3 to 4 kilohertz, and then they stop following. They don't give me a spike every half a millisecond or so. So if I give it, for example, 5,000 hertz, they cannot follow that. So what they do is you just give random spikes. And that's what is called a rate code. So basically, if this is the cochlea, and this is my 5 kilohertz location in the cochlea, then it's going to give me a random spikes because it cannot follow 5 kilohertz. And the maximum number of spikes it's going to give me is when the signal is 5 kilohertz. So if I look as a function of frequency, and I'm recording here, then it's going to give me a random spikes, but they're bigger when it's 5 kilohertz. So it sends random code, but it's maximal number of spikes when it matches its own best frequency. But if I look here at 2 kilohertz, then it does follow. It gives me one spike in a very ordered way, only when I give it 2 kilohertz, but it does not follow if I give it faster or slower. That's how it encodes information. So the key point here is that at around 3 or 4 kilohertz, we stop getting this phase-locked response that locks to the phase of the signal, and that's like a low-pass filter. And so this is what this low-pass thing does, and it has a cutoff around 3 to 4 kilohertz, because we cannot keep following at faster because the brain does not follow at faster rates. The next thing is we have this cochlear nucleus. We're trying to encode all the way up to the midbrain before we get to the cortex. So the cochlear nucleus has these very sharp responses, has this onset analysis, and the way that's done is just a simple, so has a simple derivative in spectral along the adjacent frequencies. So we do a derivative in frequency, and then we do a half-wave rectification. So this nonlinear half-wave rectification, sort of a max operation. And then the final stage is this midbrain stage, which also gives us further loss of phase lock-in. So basically, by the time we get to the midbrain, the neurons cannot follow below 1 kilohertz. So we've lost this thing that we had before. Any information that was relevant was extracted. By the time we get to the midbrain, neurons only phase lock up to 1 kilohertz or so. So we add another low pass here to indicate that we are not following information up to 1 kilohertz. And basically anything above 1 kilohertz is just energy. It's not very fine temporal information. Okay, so the next stage that we want to encode is this cortical stage. And as I mentioned to you before, the way this cortical analysis is done is with these SCRFs. If you remember, each neuron has a slightly different SCRF, encodes slightly different temporal and spectral modulations. Okay, so we can do this with a sort of a bunch of transfer functions. The only parameters that we have to care about here is we have to vary the information in time. So we have a time frequency representation. We have to vary the temporal modulations. So basically, fast versus slow. If you remember those. Let me see if I can slide through it quickly. I think that's important. You know, some neurons are slow, others are, some neurons are fast, others are slow. So we want to be able to do this in a computational model, wherever that is. Okay. So if we vary a parameter, we want to vary a parameter that controls this temporal modulation. We want to also vary a parameter that controls the spectral modulation, how wide, how narrow they are. And we want to vary the phase information. So, you know, is the impulse response, so if I look, is the impulse response this way, or is it this way? Both of these can be made to have the same modulation, but they have slightly different phases. So we want to be able to vary the phase. And the other thing we want to be able to vary is this orientation. Is it up or is it down? Oriented up or down? So now when I have a signal that I want to analyze through this cortical stage, then I want to have a collection of these neurons. However many I need, 100, 1,000. The only important thing is that I want them to vary along all these different modulations. I want to have some slow ones, some fast ones, some ones in between. I want to have some broad ones in spectrum, some narrow ones, and ones in between. I want to have ones of different phases and different orientations. So just in terms of terminology, this temporal modulation, the speed and time, we refer to it as rate, and it's encoded in hertz, because it's temporal modulations, 4 hertz, 8 hertz. The spectral modulations are referred to in cycles per octave. So just like hertz is cycle per second, then spectral modulation can be encoded in cycles per octave. Do you all know what an octave is? So an octave is just doubling of frequency. So because we're dealing with log frequency, not linear frequency, so this is frequency, right? And the brain has this log frequency, so basically 100, 200, 400, 800, because it's log frequency. So any doubling of frequency, this is 1 octave, this is 1 octave, this is 1 octave. Just like if I had the time axis, I would say it's in linear, so 100 millisecond, 200, 300. Well, I shouldn't do that. I should do in seconds. So 1 second, 2 second, 3 second. So this is linear scale. So this is 1 second, right? This is 1 second, right? So when I'm encoding temporal modulation, I'm encoding how many cycles do I have in this 1 second. If I have, say, 2 hertz, that means I have 2 cycles, right? This is what 2 hertz looks like. The same thing in the spectral domain. This is 2 cycles per octave. Well, anyway, you know what I mean. 2 cycles per octave, right? The same thing. It's just now in spectral, along the spectral axis. 2 cycles per octave, 2 cycles per second. Okay, and that's what hertz is. So, okay, so I was just telling you about terminology. So the change in temporal modulations is referred to as rates in hertz, and the change in spectral modulations is referred to as scale in cycles per octave. So I can have fast ones, broad ones, narrow in both dimensions, in addition to the phase information and the direction information. So, okay. So the way we do this is we implement a basic function in time and in frequency. The okay. So the time function is a basic exponential. Okay. So you can think of the time function as a basic exponential. The frequency function is like a Gabor function. So this is in frequency. And we want to be able to multiply them with different widths. So some narrow ones and some really broad ones with different cutoffs. So we want to be able to vary how they are. So the way we do this is this is basically a wavelet analysis. So you do this dilation along with these parameters. For the temporal, it's the rate, this small omega. For the spectral, it's the scale, the big omega. So this is, say, if I'm doing a 4 hertz versus a 2 hertz, then I would just vary this dilation. The same thing in frequency. If I'm doing 2 cycles per octave versus 8 cycles per octave, then I will just vary this parameter, the big omega. And then the next thing is I want to be able to introduce these phases. So the way we do it is we take this function now, say for the temporal one, and do this this expansion, this sort of the sinusoidal decomposition. So you have the function with the cosine of a phase plus its Hilbert transform of sine of a phase. And now I introduce a new parameter, theta. And they do the same thing for the spectral, where I can introduce another parameter for its own phase. So I'm varying the phase in the temporal and varying the phase in the spectral. Okay? All right. And then the final thing we do is we take this function here and we create a complex valued function. Now, instead of this being a real valued function, I can make it as a complex valued function by taking itself as the real part, and the complex part is its Hilbert transform. This is for the temporal one, and I can do the same thing for the spectral one. All right? And now the SCRF is just multiplying these two together and taking their real part. So the SCRF now is taking the real part of the rate function multiplied by the scale function. This is for the positive direction. And now to introduce the negative direction, we do the same thing, but this is now complex conjugate. So let me see. Just don't want to run overboard. But so you'll get to use this model later in this afternoon in the lab. The basic thing that I want you to understand is there is some expression that generates these SCRFs. And when you have an acoustic signal, what you do is you convolve these SCRFs with the signal. That's what gives you this cortical response. So if you don't follow really the math here, I'm going to put the slides so that you can look at them, since we don't have a lot of time to go over this again. But the basic idea here is we have these SCRFs, okay, that have time and frequency, all right? And we're just creating multiple SCRFs that so this is a time frequency, but they have these parameters that control them. The temporal modulation, what is called the rate. Spectral modulations, what is called scale. They have a phase, I'm just going to call it phi. And you can think of an orientation. Okay? So an SCRF, it is a time frequency transfer function that can look something like this is something like this, or it can look like, I don't know, something like really broad, or can look like something really narrow. So just by varying these different parameters, I can make it look like anything I want. Now, to create a cortical layer, I will need a lot of these with different width and different heights and different phases. So we just make a lot of these filters, depending on what we're analyzing. And now when I have the signal come in, then I want to convolve it with each one of these, right? And that's how I get a higher dimensional representation of the signal. So, right. So basically, I have my spectrogram, and each one of these is one of these SCRFs. And when you do this cortical analysis, what you do is you convolve the signal with each one of these. And that gives you a higher dimensional representation, right? Because you're convolving a two-dimensional signal, YTF, the spectrogram, with these SCRFs that are also TF, but have omega and big omega and a phase, right? So now the output is this TF, small omega, big omega, and a phase, right? So now this is a higher dimensional representation. I can sum over each one of these dimensions to look at the signal because it's higher dimensional. I can't look at it in a, like MATLAB or something. But what I achieve here is a representation that tells me what is the temporal modulation content of the signal. I can know by looking at this dimension. What is the spectral modulation content of the signal? I can look by looking at this dimension. And what is the phase of each one of these? I can look by looking at the phase information. So all these answers, I can get them by doing this analysis. But in terms of the analysis itself, it's just a basic convolution with each one of these SCRFs. OK. All right, so we're almost there. So this is the kinds of things that I told you about. This doesn't look really well, but the things that I mentioned is that these slow modulations, these cortical responses, they care about some modulations in speech signals, and these happen to match very well those in speech, for example. So this is showing you the output of this model, of this convolution, but now we sum over time and frequency and phase. We only look at the temporal modulations, the rates, and the spectral modulations, the scales. So this is for a clean signal. This is how it looks like. It has most of its energy around 4 to 8 kilohertz, and this is the positive and negative. Remember, we have the positive orientation and the negative orientation. This is not negative frequencies. It's just that, you know, I can have it look this way, or I can have it look this way. So it's just orientation. It's going upward or downward, right? So, OK. So this is analyzing a signal and showing that most of its energy is pretty symmetric. This is averaging across a lot of speech sentences. So we're canceling out a lot of the content of the speech. We're just keeping what is the modulations in this speech signal. So we know it has most of its energy around 2 to 8 kilohertz, both in the upward and the downward directions. In the spectral, it has most of its information from below 0.5 cycles per octave all the way to 2 cycles per octave. So it doesn't have anything that is as high as 8 cycles per octave. That's really, really fine frequency. I mean, if you think about it, 8 cycles per octave so is a really fine spectral information. We don't have that in speech signal. And so what you can look at is once you look at what the clean signal looks like, if I add noise to it, you see that this information gets degraded. If I add reverberation to it, basically, if you are in a real room reverberant, you have these echoes coming from the walls and the tables, you lose some of these modulations. And this is combining both white noise, just a background of white noise and reverberation. And you see how these modulations are distorted. So, so 2 things I just want to emphasize here. One is, if you have some noise that does not disrupt this pattern that falls maybe here, then you can know that it's not going to affect intelligibility of speech. It's going to be annoying in the background, like a steady tone. If I am talking and there is this steady humming, say, from the projector, you can still hear what I'm saying. It's annoying, but you can still understand what I'm saying. So what we know is that noises that don't affect the basic modulation information in speech are not going to disturb its intelligibility. But for example, very high reverberation that distorts this pattern is going to make it hard for you to understand what the signal is. So what we learn by looking at the patterns in the auditory cortex is which part of the signal really carries the information that we care about, for example, in speech. I don't have the slide here, but we can do the same thing with music, for example. Where is the tempo information carried? Where is the timbre of different musical instruments is? And you can do this analysis using this cortical model, compare how different instruments are encoded, how they're going to be integrated together and how going to be degraded if we are in a very noisy environment or something like that. So, okay. And so this is just a final slide. The main thing that I wanted to emphasize is that there are a lot of models out there that do the front-end analysis, the cochlear analysis. You can find a lot on the internet, in addition to the one I mentioned to you. The thing that I wanted to emphasize in today's lecture is that there is more information beyond just the cochlear stage. This modulation analysis is not done early on in the system. It's done more centrally. And you can use a computational model like the one I mentioned briefly.
That raster plot you showed looked an awful lot like a parallelogram autocorrelation. Is that what that?
No, it is an actual neural signal from the. It does look like a parallelogram because I chose a neuron that wherever that is. I chose a neuron that really phase locks very well. So this neuron gives very fine information that repeats. Not all neurons do that. Most raster plots are pretty messy. You just see dots everywhere. But somewhere you see some very nice encoding. So this neuron just happens to really follow very nicely a lot of modulations. Most neurons, as I was telling you, like only specific modulations. They don't like all of them. This one happens to like most of them. It follows very nicely the temporal information as we change. Most neurons would only, for example, respond to 8. They give us a spike every 250. And then for the rest, it's just random dots everywhere. So it just happens to be that way. But you're right. It looks like a parallelogram in a way because it's a very faithful neuron. It really likes whatever you give it. It just gives it to you back.
Yeah?
I have one. How do you choose these neurons to get these informations?
So you don't choose them. Typically how the experiments go, you go blindly, you put the electrode, you try to go as close to a neuron as you can by hearing just these electric pulses, and then you do the recording. And whatever you get, you get. Then you move to the neuron next door, and you do this for hundreds and hundreds of neurons. You cannot know ahead of time, because you're going blindly. You just put an electrode in an animal's head. You just know that you're recording from auditory cortex or cochlear nucleus or one of these stages. And then you move it, with micromillimeters. Sort of you're going really very finely, and you then stop at a certain point. I mean, and there are people who are experts in these things. They know that if they've gone close to a neuron, because you can hear the electric pulses being sent. And by the way, these neurons communicate with each other all the time, even if in quiet. They still send signals. So you can know that you're close to a neuron, and then you stop there, and then you start present sounds and see how the electric signals now change. And that's what this recording will look like. And then you do this transfer function analysis. Then you're done. You move the electrode a little bit next door. You do the analysis again, and then you just do it for hundreds and hundreds. And by doing a lot, that's how you know that you can vary, that some of them are slow, some of them are fast, some of them are broad, some of them are narrow. But you need lots of them for you to know that information. One of them will not be enough.
Okay.
So how many neurons are there that participate for humans?
Well, as I mentioned, the whole brain is made of 100 billion neurons. At the level of the auditory cortex, there are millions of neurons. And there is no way anybody can know what every single one does. So in these kinds of studies, you do an experiment where you record 100 to 200, another lab that does another experiment. Just people do in the order of hundreds and 200s. And then as the information gets published, then you get to consolidate it. There are other techniques, of course, that look at the whole network. There are imaging techniques. You can put dyes inside these neurons and see how they communicate and then take images. But that does not give you the fine information of every single one. It gives you the network communicating. Just like we do brain scans with humans. When you go into an fMRI scanner, what that shows is that what each part of your brain sends information, but it does not tell you what every neuron does. It just tells you that as the information came in, it went from here to this other part to this other part with a certain timing. So this is just going into the micro information. And there is more finer scale. There are people who really analyze what that specific neuron does, and they do slices of the brain and things like that. So you can do different levels of analyses, but we're dealing with millions of neurons, so there's no way of knowing, with details what each one does, but you get the big picture as different people study different parts. Okay? Any more questions? All right. Okay.